Hello friends, welcome to BAMU'S Biogenius. Today we will discuss about fertilization. This topic is for UG and PG students, so please stay with me. See, this is the first slide about introduction. We will discuss history of fertilization, then we will discuss about structure of a ovum, then we will discuss about sperm, then steps in fertilization. First point is deposition of sperm in female genital tract. In this, we will discuss about composition of a semen, what is a seminal fluid and prostatic fluid. Then next point will be capacitation of a sperm. Then third point will be recognition of zona pellucida. Then acrosomal reaction. The next point will be how the membrane fuses with each other. Then how block to polyspermy occurs. There are two blocks, first fast and second one is a slow block to polyspermy. Then the last topic of fertilization is fusion of male and female pronuclei. So let us start with the lecture. As far as history is concerned about the fertilization, Oscar Hertwig and Hermann Fold were the first scientists who observed entry of a sperm into egg and fusion of a nuclei in sea urchin eggs. This was happened in 1876. So the fertilization is a union of a sperm and egg to produce a zygote. So this is the smallest definition of fertilization. Now we will discuss about the structure of oocyte. The structure of oocyte has certain peculiarities that we need to discuss here. See, the first one is the size difference. The sperm and ovum or oocyte is different in size. In case of humans, if we compare the sperm and ovum, the sperm is about 65 micrometer in length, but the oocyte is 0.1 mm in diameter. So it is a 10,000 times larger than the sperm. Now, why such a kind of a drastic difference in size is there? That is because it that oocyte accumulates cytoplasm. We know that during meiosis, a phase of growth is there, and in this phase of growth, the oogonial mother cell undergoes this growth phase and becomes primary oocyte. And this primary oocyte undergoes a division, first meiotic division, that is called as M1, and a secondary oocyte is formed with a very small polar body. That means whatever the accumulation of cytoplasm is there, that is conserved in the daughter cells. Again, in M2, the same division is observed, that the polar bodies are formed. Again, a secondary polar body is formed and the oocyte is formed. So this is how that the conservation of cytoplasm is there in case of oocyte, but you will not observe such a kind of unequal division of a cytoplasm in case of a sperm, where in case of a sperm the four daughter cells which are formed, they are of the same size. So this is the difference, and that is why the cytoplasm is more, the size of the oocyte is larger than the sperm. This cytoplasm, accumulated cytoplasm, contains various types of materials, and these materials are very useful for the development of the zygote after fertilization. What kind of a material that is? The material is, for example, nutritive proteins are there. We know that the nutritive proteins are required by the zygote after fertilization, because this zygote does not gain any kind of energy or a food material from the surrounding medium. So until it is placed in the uterus in case of mammals. So for that particular period, it requires energy, and this energy is provided by these nutritive proteins. Along with that, certain kinds of amino acids are also become available due to the nutritive protein that are present in the cytoplasm. Then another material that is very important for a cytoplasm is ribosomes, even transfer RNA also. Now see, when this particular zygote is formed after fertilization, this unicellular zygote is converted into a multicellular organism, and when they are producing many cells from a single cell, number of material, or we can say that proteins, structural proteins are required, the enzymes are required, and for the synthesis of these structural proteins, enzymes, ribosomes and transfer RNAs are very important, and that's why these ribosomes are present in the large amount in the egg cytoplasm. Then these ribosomes, now what is the raw material for a ribosome? Raw material for a ribosome is a ribosomal RNA. From where this ribosomal RNA is synthesized? Ribosomal RNA is synthesized from ribosomal DNA. Now, in case of amphibians, you will see that lamp brush chromosomes are there. These lamp brush chromosomes are responsible for synthesis of ribosomes, and these ribosomes, or we can say that these ribosomes are synthesized from a ribosomal RNA, and these ribosomal RNA are synthesized from the ribosomal DNA. Now this ribosomal DNA is amplified so that it can produce number of ribosomal RNAs, and these ribosomal RNAs then can give rise to a ribosomes. Similarly, transfer RNAs are also formed. So these are molecules, or we can say that these structural elements that are present in the oocyte. Then messenger RNA is also there. It is called as a informosomes. It is in the latent condition. It is not expressed of till fertilization. After fertilization, it starts to work. Now, this messenger RNA contains certain kinds of information for a synthesis of a particular specific type of a proteins, and that's why these mRNAs are called as informosomes, and these are present in the oocyte cytoplasm. Then morphogenetic factors are also present. Now, when we know that the morphogenetic factors means what? These morphogenetic factors are nothing but the kind of transcription factors or proteins that govern the synthesis of a particular type of a protein from the DNA, and these morphogenetic factors are responsible for a differentiation of a cell. Now, when this zygote becomes a multicellular animal, the morphogenetic factors that are present at the particular region of the oocyte that are affecting the development of these particular cells, and thereby producing a differentiation in the that multicellular zygote. So these morphogenetic factors are also present in the cytoplasm. These morphogenetic factors are also responsible for example, these are responsible for formation of anterior and posterior axes, dorsal and ventral part of the embryo, like that. So these morphogenetic factors are playing important role in a oocyte, and that's why they are present there. Then the protective chemicals are nothing but the chemicals that are present in the cytoplasm. These protective chemicals makes that particular egg unpalatable for the predators. In case of external fertilization, the eggs are laid on the ground and they are exposed to the UV radiations. To avoid the damage, DNA damage due to the UV radiation, the cytoplasm of the oocyte contains uh, DNA repair enzymes, and these DNA repair enzymes, um, even if there is a damage, this DNA repair enzyme, they protect the DNA. So along with the cytoplasm, so this is all these special molecules, or we can say the factors, proteins, ribosomes, all these are meant for proper development of the zygote, and it increases the survival value, or we can say the probability of a survival of that particular zygote. Along with that, nucleus is also present. Nucleus is in the form of a pronucleus. So these are the most important ingredients that are present in the cytoplasm. Now, externally, the this is if we see the structure of a human egg, the cytoplasm is bound by the oocyte plasma membrane. Outside oocyte, you will find a space. This is known as a perivitelline space. Now, this perivitelline space contains a polar body. As we know that the human egg is a secondary oocyte, and that's why it contains the polar body. Then outside this perivitelline space, uh, glycoproteinaceous layer that is present. It is called as a zona pellucida. After zona pellucida, the layer of corona radiata cells are there. The corona radiata cells are nothing but the cumulus cells, but they are in direct contact with the zona pellucida, and that's why they are called as a corona radiata cells. After corona radiata cells, the cumulus cells are also present around the egg, and around this cumulus cells, you can see here a layer of a connective tissue is there. This layer of a connective tissue is made up of polymers of a hyaluronic acid. So this is how that this particular egg is ovulated from the ovary and then taken to the ampulla region of the um, you can say the uterus, uterine tube. So this is all about the structure of a oocyte. Now we will discuss about the structure of a sperm. See, when we talk about the structure of a sperm, first thing that comes in our mind is its size. The size of the sperm is, in case of humans, is about 65 micrometer in length. Now, it is divided into the head region, then a mid piece, which is called as a mid piece, and the tail region. The last part of the tail is called as a end piece. Now, if we see the head region, or you will find that the whole sperm is covered by a plasma membrane. Inside a plasma membrane, in the head region, we will find the two-third of the region of this large nucleus is covered by a acrosomal vesicle. This acrosomal vesicle is derived from a Golgi complex, and it contains hydrolytic enzymes. It is a membrane-bound vesicle and contains hydrolytic enzymes. These hydrolytic enzymes helps in fertilization. So inside this, or inside the head region, the nucleus is present. Nucleus does not have a nucleoplasm but a compact mass of DNA is there. This nucleus is made up of the compact DNA material. This compaction of DNA material is achieved by replacing histone proteins with the protamines. These protamines are arginine and cysteine residue rich proteins, which we know that arginine is a negatively charged, highly negatively charged, and because of that, it makes this protamines more basic, and because of that, uh, because of the cysteines that are present in the um, this protamines, they form sulfur bonds, and these again the sulfur bonds helps in compaction of the nuclear material. So this is how that this nucleus is a highly compact material or region which is present at the head of the sperm. After that, a mid piece is there. This mid piece contains centriole and mitochondria. These mitochondria are responsible for providing the nutrition. Sorry, these mitochondria are providing the ATP to the um, these axial filaments so that these axial filaments can move. So these axial filaments contains a 9 plus 2 arrangement of microtubules. These 9 plus 2 arrangement of microtubules helps in movement of these axial filaments, and because of that, the the this sperm can move forward. Then if we consider the percentage of motile sperms in a ejaculate, there are about 80% of the spermatozoa are motile in a ejaculate. Then the movement is about 5 mm per minute inside the vaginal tract. The survival rate, or we can say the of time period of these sperms is about 3 to 4 days. Within a 3 to 4 days, these sperms will die. So this is all about the structure of a sperm. Now we will discuss about the steps in fertilization. First step in fertilization is the deposition of sperms in a female genital tract. See, the sperms are deposited in the female genital tract with the help of a semen. Semen is nothing but a kind of a fluid secreted by 5 regions of the male genital tract. First region is the testes, where the sperms are synthesized. Then epididymis, where sperms are stored. Then seminal vesicles, which secretes a kind of a fluid that is known as a seminal fluid. Then fourth region is prostate gland. This is a prostate gland produces a prostatic fluid which accounts about 20% of the semen. The prostate gland is of the size of the small apricot. This gland is responsible for secretion of prostate specific antigens, and these are responsible for liquefaction of the seminal fluid or a semen. This PSA is also important in detection of cancer of prostate gland. It has been found that the prostate gland interferes in the urination also in elderly people. So it has also been found that most of the elderly people who are above 40 and 50, they experiences itching in a genital tract. That is because of the increase in the size of the prostate gland. The most of the people, they suffer a prostate cancer, and that prostate cancer is also of this prostate gland. Then the last one is bulbourethral gland. This bulbourethral gland produces a bulbourethral fluid that amounts to 5% of the semen. Bulbourethral gland also contributes the bulbourethral fluid, and this bulbourethral fluid contains mucoproteins, which are very important ingredient of the semen. Now, each of these glands, they have a separate kind of chemicals that are poured in a semen. Seminal fluid is a kind of alkaline fluid, and it helps in survival of the sperms in a acidic environment of the vagina. Seminal fluid also contains prostaglandins. It also contains the fructose. The prostaglandins helps in carrying out or stimulating the vaginal tract for contractions. Because of that, it aids in movement of a sperms towards the uterus. Then the pros after prostaglandin, the fructose that is present in the seminal fluid, that helps in giving energy to the sperms, because these sperms absorbs this fructose, and from this fructose the mitochondria produces ATP, which is utilized in the movement of the flagella, or we can say the tail of the sperm. Then prostatic glands, these prostatic glands are responsible for the secretion of a prostatic fluid that contains phosphatases, then proteolytic enzymes, which is responsible for the first clotting of the sperms and then its liquefaction. The proteolytic enzymes aids in liquefaction of the semen inside the vagina, so that the sperms can be released from that particular clot. Then bulbourethral gland. These bulbourethral glands are responsible for the secretion of the mucoproteins. These mucoproteins are important ingredients of the semen. After deposition of these sperms, or we can say the semen, inside the uh, vaginal tract of the female, the sperms moves towards the uterus. Now here, the in a vaginal tract, the environment is acidic, but as the semen is alkaline, it makes or it neutralizes the effect of acidity in this particular portion. Then this region of the uterus is called as a cervix. Now these sperms moves upwards towards uterus through this cervical region. It is guarded by the mucopolysaccharides, fibers of a mucopolysaccharides. It contains a mucus, and through this mucus, the sperms migrate to the this particular portion of the uterus. Now here, this region is a very alkaline, and it is a conducive for the movement of the sperms. So here, after coming in this region, the sperms intensifies its movements and it moves towards the opening of the orbita. Now you will find that after some time, these sperms enters into the isthmus region of the fallopian tube. This is the isthmus region of a fallopian tube. about 1% of the sperms deposited in the vagina, only 1% of the sperms uh, are able to come to the stomach region of the fallopian tube. From this stomach region, then sperms moves towards the ampullary region, where in this ampullary region or ampulla of the fallopian tube, the sperms they fertilize the ovum. So this is a deposition of a sperm in a female genital tract. Now you can see here what is the time that it takes from the vagina to reach up to the ampulla region is only 30 minutes. So within 30 minutes these sperms move towards the ampulla region. Now the second point is the capacitation of the sperms. As I have told you that within 30 minutes, within half an hour, the sperm will reach up to the ampulla region and to fertilize the ovum. But these sperms, those who reaches within half an hour to the ampulla region to fertilize the ovum, will not be able to fertilize the ovum. It has been found that even if we extracted sperm from the epididymis and placed on the ovum, still these sperms will not be able to fertilize the ovum. It has been found that the sperms need to spend some time inside the female genital tract so that they can get the ability to fertilize the ovum. Now, this gaining of ability to fertilize the ovum after certain kinds of biochemical reactions inside the female genital tract is known as a capacitation. Or, in other words, we can say that the sperms required to spend certain time inside the female genital tract so that they should be exposed to the environment inside the female genital tract so that certain kinds of chemical reactions that should take place, and these chemical reactions enable sperm to fertilize the ovum. So it is nothing but the kind of a capacity building of sperms, and these after this the sperms can fertilize the ovum. Now what kind of these biochemical changes that takes place inside the sperm, or we can say that uh, they brings about the capacitation of a sperm. See, in even in vitro conditions, whenever a person goes for IVF, the sperms need washing, and this washing means what? This washing of the sperm means here also a kind of biochemical changes that takes place inside the sperm. Now see, the first biochemical change that takes place inside the sperm when the sperms are placed in a female genital tract is the cholesterol efflux. Now, efflux means the uh, removal of cholesterol from the sperm plasma membrane. So, it has been found that the sperms, when placed in a female genital tract, the cholesterol is removed from the membrane of the sperm. Now, if we see the content of the plasma membrane of the sperm, it has been found that the cholesterol is about 25% in the plasma membrane. The 70% of the um, plasma membrane is made up of phospholipid, and 5% of the plasma membrane is made up of carbohydrate. The ratio of cholesterol to phospholipid is very crucial in for a capacitation. See, if cholesterol is more, the sperm has to spend more time inside the female genital tract for a capacitation. So, this when the sperms are placed in a female genital tract, the cholesterol is removed. Now, There is a mystery in this, that how this cholesterol is removed, what kind of a molecules, what kind of a chemical reaction, or what kind of a binder that is present in female genital tract that extracts this cholesterol from the sperm is not known. But when in vitro conditions, it has been found that uh, albumin that is helpful in extracting cholesterol from the sperm. So whenever there is a treatment of IVF, during the IVF, sperm washing is done, and that in that sperm washing, the albumin is used. See now, when the cholesterol efflux is there, that means cholesterol is removed from the plasma membrane. The membrane fluidity is affected, as we know that in any kind of a plasma membrane, when cholesterol is present, it gives a kind of a stability to the plasma membrane. But when this cholesterol is removed from the sperm plasma membrane, the membrane fluidity is affected. This is known as a remodeling of the sperm plasma membrane, and because of that, certain kind of changes that takes place in the sperm plasma membrane. Now, because of this, the K positive ions inside the sperm, they are removed, or they goes out of the sperm, and because of that, the membrane potential changes. As this membrane potential changes, the certain kinds of channels that opens. For example, in this case, the calcium ions and bicarbonate ion channels that opens, and because of that, the calcium present in the surrounding medium inside the female genital tract that enters inside the sperm. Even bicarbonates that are present in the seminal fluid that also enters inside the sperm. Now, because of that, we can see here the pH of the sperm that increases. See, the calcium ions are entering, bicarbonate ions are entering inside the sperm. So naturally, what will happen? The pH will increase. Now, this increased pH is helpful in activation of a protein kinase A. And it has been found that whenever a protein kinase A activation takes place, it produces the ATP, and this ATP gives rise to a cyclic AMP, and this cyclic AMP then induces, or we can say that it helps in rigorous movement of flagella or tail of the sperm. That means, due to all these kind of a chemical reactions, what is happening? The hypermotility of a sperm is taking place. Now, along with that, there is a role of calcium also. When calcium enters inside the sperm, it has been found that this calcium is helpful in bringing about the, the acrosome reaction, because we know that a membrane fusion is there, and that membrane fusion is always mediated by the calcium. So, the acrosomal membrane and the membrane of the plasma membrane, the sperm, is fusing at that particular point when the calcium level rises, and because of that, the content of the acrosome is poured outside the sperm. So, this is the role of a calcium. Now, another role is that because of this, because of all these opening of the channels and all that, the intracellular alkalization of a sperm that also takes place, and that also helps in acrosome reaction. Then role of progesterone hormone is also there. A small amount of a progesterone is secreted by the cumulus cells, and this progesterone, the sperm has a kind of receptors on that, and these receptors for this progesterone, and when progesterone comes and binds to these receptors, it has been found that the motility of sperm increases, or it stimulates the motility of the sperm. So the sperm becomes hyperactive, and they fuses with the, or they start to move towards the more vigorously towards the ampulla region. So you can see that all these different kinds of chemical reactions that are taking place inside the sperm, and that enables the sperm to move towards the ampulla region and fertilize the egg. So for these all these reactions, a time is required, and in that particular period, this capacitation takes place. Now, as I told you that although total female genital tract is responsible for a capacitation of a sperm, but the isthmus region is more important because it temporarily stores the sperm, and when the sperms are deposited in the vagina, only 1% of the sperm can enter inside the fallopian tube, where these sperms in the isthmic region or isthmus region of the fallopian tube, these sperms are stored for some period of a time. These sperms attaches to the walls of the isthmus region, and where the only capacitated sperm that can leave this isthmus region. Now, why these sperms are capable of release itself from the walls of the isthmus? That is because of the vigorous or increase in the motility of the tail of the sperm or capacitated sperm. The sperms are able to um, move towards the um, ampullary region, and we can say here that a kind of temperature gradient is there around during the uh, around the ovulation time. So this temperature gradient attracts these capacitated sperm towards the ampullary region, and when these sperms reach up to the ampullary region, chemotaxis is also there, chemicals secreted by the cumulus cells that attracts the capacitated sperm towards itself. So this is how that the sperms are taken towards the ampullary region for fertilization. As I have told you that the third step is a recognition of a zona pellucida. See, a layer of zona pellucida is made up of three types of proteins. These are known as ZP1, ZP2, and ZP3 proteins. ZP1 proteins are responsible for attaching the ZP2 and ZP3 monomers. Now, this ZP3 contains a kind of residue that is known as an N-acetyl glucosamine residue, and this residue is responsible for attachment of the sperm to the zona pellucida. Now, see the GALT receptors which are present on the sperm plasma membrane. They are uh, they interact with the this acetyl glucosamine residue on a ZP3, and they get bind to the zona pellucida. Now, these GALT receptors are exposed due to the capacitation reaction, and because of that, these GALT can interact with the acetyl glucosamine. So that's why it is necessary. Capacitation is necessary to interact with the zona pellucida layer. Now, as soon as this GALT interacts with the um, N-acetyl glucosamine residue on the ZP3, kind of in its intracellular domain get activated, and the intracellular domain stimulates the G protein. Now, because of the stimulation of a G protein, the further opening of calcium ion channels that takes place, and because of this increase in intracellular calcium ions, there is a fusion of the acrosomal vesicle and sperm plasma membrane. So, at various points, multiple points, this fusion of the acrosomal membrane and the sperm plasma membrane at the head region that takes place, and because of that, the acrosome reaction that starts. Now, the fourth point is acrosome reaction. See, as the sperm head binds to the zona pellucida, I have told you that calcium ion channels that opens and it increases the concentration of calcium inside the sperm. Because of this, the membrane of the acrosomal vesicle that fuses with the membrane of the head region of the sperm, and this fusion at multiple points makes the release of the acrosomal content outside the plasma membrane of the sperm. And when it comes out of this acrosomal vesicle, this hydrolytic enzymes they digest the zona pellucida, and because of that, a kind of a tunnel is created in a zona pellucida so that the sperm can enter or penetrate inside the zona pellucida. So you can see here the acrosomal content that are poured, and the region of the zona pellucida is digested, and a kind of a tunnel is created, and through this tunnel the sperm enters or penetrates inside the um, zona pellucida and reaches up to the perivitelline space that is present in between the plasma membrane of the oocyte and zona pellucida. So this is about the acrosome reaction. Now see what is the effect of acrosome reaction. A part of inner membrane of the acrosomal vesicle fuses with the um, membrane of the plasma membrane of the sperm, and because of that hitherto those proteins which are hidden inside the acrosomal vesicle are now exposed to the external environment, or we can say they are exposed in a perivitelline space. Now the fifth point is membrane fusion, where the acrosome reacted sperms they uh, exposes certain kinds of hidden proteins. These proteins are known as a fusogen. So this is a fusogen. So these hidden proteins, fusogens, are called as a isomo-1, and they interact with the membrane protein of the oocyte that is known as the CD9. So these CD9 are the proteins or membrane proteins that are again distributed over the plasma membrane of the oocyte. So this isomo-1 and CD9 interact with each other, and they are now responsible for fusion of the egg plasma membrane with the sperm plasma membrane. This is how the fertilization takes place. You can see the sperm approaches, then it reaches to or binds to the zona pellucida, recognition of a zona pellucida, then the acrosome reaction that takes place, and the acrosomal content is poured, then it makes a kind of a tunnel through which the sperm enters inside, then uh, this sperm after acrosome reaction it reaches to the plasma membrane of the oocyte, and then fusogenic proteins they are responsible for fusion of sperm plasma membrane with the oocyte plasma membrane. So this is known as a membrane fusion, and the male pronucleus enters inside the oocyte. So along with that, the centrioles that also enters inside, and a part of the flagella that also are a part of axonomy that also enters inside the cytoplasm of the oocyte. And you can see here the plasma membrane of the sperm that is dangling on the surface of the oocyte. If we see the case of a mice, it has been found that protein which is present on the plasma membrane of the sperm that is known as a fertilin interacts with the protein that is present on the plasma membrane of the egg. That protein is known as alpha 6 beta 1 protein. That two interacts with each other and they fuses with each other. So they helps in fusion of the membrane with each other. It has been found that the mice homozygous for the mutant fertilin is unable to fertilize or unable to fuse with the membrane of the egg plasma membrane. This is about the fusion in case of mammals, that is mice. You can see after membrane fusion, the sperm nucleus, then mitochondria, centriole, and the flagellum can be inserted inside the cytoplasm of the uh, female gamete, and thus the fertilization is accomplished. Now the next point is block to polyspermy, that is also called as a zonal reaction. In case of mice, it has been found that, or in case of humans also, it has been found that when the membrane fusion takes place, the other sperms are not allowed to enter inside the egg or fuse with the egg plasma membrane. Now, this kind of um, we can say the mechanism that is known as a block to polyspermy, and it prevents the entry of more than one sperm inside the egg cytoplasm. Now, this is accomplished with the help of the proteins that are secreted by the cortical granules. These proteins are responsible for cleaving the N-acetyl glucosamine residue on ZP3. And when these cortical reaction takes place or a zona reaction takes place, the whatever the acetyl glucosamine residues that are present on the ZP3 that are cleaved off, the attached sperms which are um, present on the zona pellucida, they are also then uh, detached from the zona pellucida, and even there is a change in the ZP1 and ZP2 um, proteins that are present in the zona pellucida. So this is how that block to polyspermy is achieved, and only one sperm is allowed to enter inside the female gamete. The last point is the fusion of male and female pronuclei. When sperm enters inside the oocyte cytoplasm, at that time the oocyte undergoes a second meiotic division and forms a second polar body and the ovum. Now, this sperm which enters inside the body, the male pronuclei of that sperm decondenses and increases in size. Now, at that same time, both of these male and female pronuclei undergo DNA replication, and now they have a pair of chromatids in each chromosome. So, at this point, they become indistinguishable, and this is how that the centriole that is inserted by the sperm inside the female cytoplasm, that helps in the formation of total zygote which is capable of division. So this is how fertilization is accomplished. So this is all about fertilization. Please subscribe to my YouTube channel, BAMU'S Biogenius, for latest uploads, or like my videos and comment. Before going, please scan this QR code for fertilization quiz. Quiz number 9. Thank you.